Okay, it is now time for our third lecture of this chapter. This section being section 2, or 1.2, I should say specifically, the Chemical Foundations chapter. You are chemistry majors, so you are well aware of all the intricacies and what's going on with the periodic table. You've covered this, Gen Chem 1, 2, 3, organic by now, maybe some of you even inorganic. What you need to keep in mind is that when it comes right down to it, the majority of the elements you are going to see in this periodic table have absolutely nothing to do with organic chemistry. If they have nothing to do with organic chemistry, then they have nothing to do with biochemistry. Because remember, in biochemistry here, we are dealing pretty much only in organic molecules. An organic molecule is going to be a molecule that is based around carbon. Now, many of these other elements you see here will form compounds with carbon. The majority of them you're going to see shown here in, I don't know, what is that, a salmon coloration? Hydrogen, sodium, potassium, calcium, nitrogen, oxygen. Okay? These bulk elements are going to form literally the majority of the structures that we will find in the amino acids, the nucleotides, the lipids, and so on. But it's those yellow ones you're going to find either bound to or associating with an organic molecule. These are going to be the things that are going to be found in the cofactors that aid in the activity of the enzymes, going to aid in the correct structure formation of one of these biomolecules. Because remember, I've said in the previous lectures for this chapter, structure dictates function. You don't have the right structure, you don't have a function. And if you had me, or for Cell and Molecular downstairs in biology, you will remember I've always discussed knowing and being aware of the functional groups. Because the functional groups are what give the carbon chains, which is literally what amino acids, nucleotides, lipids, and all these, are. Carbon, 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 carbon linked together in three-dimensional structures. But the thing is, the carbon chains themselves don't do anything. They will form covalent bonds with something, yes, but that's it. What allows these carbon chains to interact with other things, other atoms, other molecules in their environment are these functional groups. Carbon is an exceptional atom, okay? There is a reason why every living thing on this planet is based around carbon. We are organic life forms. I always make the joke, if you're, a sci-fi nerd like me, you've seen it in Star Trek all the time where they say it's a carbon-based life form. We are organic life forms. Carbon. And when you look at the different bonds that carbon can make, whether you looked on the left or you look on the right, the Lewis dot molecule, or I prefer those nice lines, so much easier to see. Carbon making single, double, triple bonds with other atoms. And it's because of its 4 openings in its valence shell that it's able to form so many different bonds, different bond types, with many other atoms. It's because of this empty 4 spaces in its valence shell that it forms, as you see here on the far right or far left, an A, that tetrahedron. That tetrahedron is the shape that a carbon-based molecule will form. Shape dictates function. Its shape determines how it's going to interact with other atoms, other molecules in its environment. B shows you a carbon-carbon double bond. Basically what you end up with is 2 tetrahedrons inverted with each other. That's the singular bond. What's interesting though, if it's a singular bond, it's inverted tetrahedrons. But if it's a double bond, you now get a plane. Shape dictates function. The change in the shape here is going to be due to the number of carbon-carbon bonds. Notice the bond angles. The bond angles are very important. Even though it is a carbon-carbon single bond, as you see there in B, the angle between whatever's bound to the carbons is still going to be maintained at 109.5 degrees. But the minute it's a double-double bond, you now end up with 120, as long as everything stays in the same plane. And that's going to be a big thing, as we're going to notice. Hopefully you've seen it and you remember from organic chemistry. You know, you have the plane, but are the other atoms going above, or are they pointing down below the plane of the carbon-carbon bond? And then when you go and you start looking at the functional groups, what's attached, how they are attached, you start to begin to see some extreme differences in things. I mean, difference, hopefully you remember from chemistry, methyl, ethyl, carbon-carbon, single carbon, double carbon. They are basic. But you start adding in sulfur bonds, sulfhydryl. Get a sulfhydryl R group on a carbon chain. You can now start to form, you know, as you see here in the bottom right, disulfide bonds between neighboring R groups. It doesn't even have to be neighboring R groups. It can be R groups at different ends of that carbon-carbon chain, whether that carbon-carbon chain happens to be in an amino acid chain, happens to be, you know, some form of nucleic acid based thing, or lipid, or whatever. And as you go and you start adding in all of these different chains, you begin to see that, you know, the carbonyl, the carboxyl, the hydroxyl. Ooh. Start to see where things can now be added, subtract rather easily, gained and lost, charges showing up. Acetyl coenzyme A. Look at all of this is going on here. Now, yes, you notice that there's lots and lots of carbons. Carbon seems to primarily what everything is based on. You start up there at the nucleotide base. It's a you start going around there and you see, yes, there are some nitrogens interspersed here and there within the chain, but it is primarily carbon-carbon. Single carbon, double carbon bonds. But the carbons themselves, as I said, cannot interact with anything. What can? Notice all those places you see around here that have an O negative. Oh, yeah. So we're going to see that's going to open up for the possibility of hydrogen bonding with something. The hydroxyl you see halfway through there. Yeah, slight change in pH, and that's going to be an O negative hanging out there. More hydrogen bonding potentiality. The double oxygens, carbon double oxygen bonds you see down through there. The carbonyls, the carboxyls, and all of this are going to allow this organic molecule to interact with other molecules. Sorry, had a brain freeze there. So you go and you start looking at metabolisms. You start to see that there are literally, you look at prokaryotes, eukaryotes, and all the plethora of the different eukaryotes. Protists, fungi, animalia, plants, whatever. There's quite a bit of commonality. There are common sets of amino acids that are going to be found in prokaryotes and eukaryotes. The nucleotides are the same. The majority of the sugars and things that we use in their phosphorylated derivatives, the same. But it's when those start to get a little tweaks and twists, that's where you start to get specificities to fungi, specificities to plant, specificities to animalia. And you start digging in and you're looking closer and closer at these secondary metabolites, methylated, ubiquinations, different little things that can be added to, slight tweaks to things. Well, now instead of just being for animalia as a whole, it's for insect, it's for mammalian, it's for. So yeah, honestly, when it comes right down to it, somebody could go through and take some living tissues and grind it up, leaving fragments, no living cells, no intact cells, hand it to you as long as you have a really good setup in the biochemistry lab. You could go through and start not only saying, okay, here's the nice long list of all the things that are universally there, all the different amino acids, all the different nucleotides and things like that. And then you start looking at, what's not here. These secondary. How was the DNA methylated? How was the what was the post-translational modifications found on the proteins? Which different phospholipid derivatives were present? And you can start shrinking that list of possible organisms. Keep in mind, the macromolecules, these biomolecules that we're going to be talking about, are huge. Okay? When we're talking proteins, nucleic acids, the polysaccharides forming celluloses and things like that, they are huge. Okay? Some of the proteins are going to be anywhere from hundreds, 100, 200, up to 5, 6, 7, 10,000 amino acids in length. The nucleic acids, oh my gosh, as I've talked about in a previous lecture here for this chapter. The nucleotide count in just your chromosomes is going to be measured in the billions with a B. The bacteria in you, its nucleotide count is going to be measured in the millions, M. So when you look at proteins, structure dictates function. They have to have specific structures, to have specific functions. Primary, secondary, tertiary, quaternary. Any changes to primary could impact secondary, tertiary, quaternary. Some change, if not a complete loss, to function of that enzyme, that protein. Nucleic acids, A, G, C, T's. T means DNA. A, G, C, U. U, not a T. That means RNA. Deoxyribonucleic acid, ribonucleic acid. Shape dictates function. How do our cells know whether they're dealing with RNA, DNA? Well, is that oxy there? And where are there U's or T's present? Then you look at the polysaccharides. All living things have polysaccharides. Okay, polysaccharide just means many sugars. It's how many sugars, what type of sugars, what are the bonds of those sugars, that glycosidic linkage, alpha, beta linkages, what's the length, what side R groups or any functional groups have been added. That will determine the function of that polysaccharide. And then you got the lipids. Lipids are, insoluble, water-hating, hydrophobic compounds. We kind of group everything together. Hope you remember that from Cell and Molecular downstairs in biology. So when you go and you start looking at the lipids, membranes, lipids, a lot of ways to store energy, the fats, lipids. So when you go and you start looking at all of these, keep in mind, if you look up here at A, the A here, the amino acids. All amino acids have a set structure that will be found through all of them. Alpha carbon, the carboxyl, and the amino group, hence amino acid. The carboxyl group brings in the acid part. The amino group brings in the, as I said, the amino. You go and you look at here at B, nucleic acids, ribose, deoxyribose. Pretty much it's going to be the same for all things. Here they're showing the U, the A, the G, uracil, adenine, guanine. They're not showing is the cytosine or the thymidine. If U's present, it's RNA. If T's present, it's DNA. And when you look at a living organism, we're going to go for a nice basic one, E. coli. And you're going to see that's a theme throughout many chemistry, biochemistry, biology textbooks. We keep referring to E. coli. Why? Because we've studied the hell out of it. We have characterized this so much. So you go through and you can see just how much is there, how much of all of this. Water is going to be the primarily what is there. But you go going down, proteins, DNA, RNA, polysaccharides. What's interesting though is if you look at this far right column, 3,000 different proteins present over 3,000 different molecular species of RNA. That means every one of those proteins had to have its own mRNA. Those mRNAs, going to be slightly modified, give you different proteins under different considerations. Some of those RNAs are going to be for ribosomal, some of them are going to be for, have other metabolic functions other than just mRNA, because you got to remember you have the mRNA, the tRNAs, and the rRNAs. I've said this phrase, structure dictates function, many times in this lecture. One of the things we have to take into account is, when we're talking structure, the configuration, which atoms are present, and what is their spatial arrangement. Are they above or below a plane? Are they pointing in directions? What directions are they pointing? Because some slight modifications, pointing this way versus pointing that way, doesn't sound like much of a difference, but it's a change in structure. Change in structure, change in function. So we have to take into account stereoisomers. That on paper, the formula, the structure may look the same, but a slight modification of which direction something is pointing, which carbon it's attached to, will have major impacts on its overall structure, thus on its overall function. You look at A, to the far left here. You'll notice that instead, some of the bonds, carboxyl up at the top, C, double bond O, single O, just like the methyl at the bottom, CH, CH. It's to tell you, since it's just a line, that they are in the same plane. They are going with the normal rules that we would see with carbon molecules. But as a refresher, you'll notice that there are, here at the central carbon, radiating off the central carbon, are two different pie-shaped arrangements. The jagged line and then the fully filled in black wedge. The jagged line wedge going from the central carbon to the carbon above it. Hopefully remember from organic, that is to tell you that this is a carbon that is going back, away from. Whereas the filled in wedge shape is telling you that the hydrogen is coming out, coming towards. Filled in wedge coming towards, jagged going away. And it's a little hard to see as a ball and stick here at B, but it's a little more plain to see here in the space filling model. Black spheres are carbon, red spheres are oxygen, gray ones are hydrogens, blue sphere is nitrogen. You can see the central, right there, you can see the central carbon. The jagged line wedges telling that those other two carbons are behind. The wedge, filled in wedge, saying is that nitrogen is coming out, coming towards. We have to keep this in mind because when we go and we draw structures, as you see here with malic acid, we draw it out with the lines, and here, single lines, and, single covalent bond, double lines, double bonds, blah, blah, blah. But it's hard for our brains to take into account that some of those atoms in that molecule are pointing out, some are pointing back, as you can see in the ball and stick models. This becomes something that's also important when you're looking at double bonds, as you see there with the 11 cis retinal versus the all trans retinal. But taking into account the fact that that double bond, remember, forms that plane, is the remaining carbon chain to the right of the double bond, pointing down, pointing up. Change in shape, change in function. Because we have to keep in mind that for a lot of these molecules, especially the smaller ones, it's not set in stone. This is the shape you're going to have. There's going to be some rotation, some movement of the atoms within. They still are going to maintain the overall angle, bond angles, because they are, still going to be moving. Things are going to change, but there's a limited parameters on how much they can move about. Take into account chiral centers. Here we're not talking about the free movement of the atoms within the molecule. Here we're talking about the generalized shape, where things are in relation to each other. If you look at the far left here, carbon with atoms A, Y, B, X. A, Y, B, X, there will be some modification, some movement of. When you look, it moves. The atoms in the molecule can rearrange to a certain point, and now you have A, X, Y, B. All the atoms are still there, but they've kind of switched. Who's closer to who? Doesn't sound like a big difference, but it's a complete change in shape. Change in shape, change in function. Biochemists have a big problem, just like organic and inorganic chemists have, with enantiomers. You go through and you devise the perfect reaction. Gives you the final product that you want. All the right carbons, nitrogens, phosphates, whatever are present. But are they pointed in the right directions? You look at the two you see here. Central carbon chain, 4 carbons. Okay, all looks good there. You have some R groups, X's and Y's. Ruh-roh, Shaggy. Far left, the enantiomer shows XY to left. It's enantiomer XY to the right. Oh, that's not a big difference there, Bubba. It's a major difference. The shape will be reminiscent, but enzymes do not recognize substrates that are reminiscent of the correct shape. Cell receptors do not recognize, as you see to the right here, XY, YX. Change in shape, change in structure, receptor's not going to recognize it. So you now have spent however many months and however many millions of dollars designing, developing something that's not going to work. So what we see is we have to come up with a way to discuss shape, how things are, especially with enantiomers. You'll see this in a lot of biochemical as well as biological papers when they're discussing different substrates, different compounds. R something, S something. It's the orientation of the functional groups around a certain carbon. Which way are they going? Are they circling this way? Are they circling that way? And you see there is a hierarchy in how you go and prioritize things. Is it clockwise? That's R. Is it counterclockwise? That's S. And what's interesting, as I mentioned just a few slides ago, the shape of a molecule can be variable. It will contort. It will move around within the limitations of the bonds and the interaction of the R groups within that molecule. They refer to this as conformational changes. It will move and it'll go back. It'll move and it'll go back. It'll move and it'll go back. This torsion around that carbon-carbon single bond allows it to change. And it does it in a certain period. This way, then back. This way, then back. When we go and we start looking at biomolecules in this need for specificity, a great example is here is that big blue blob you see there is a 3D representation of the topographical representation of an enzyme. Don't know what enzyme it is. Don't care what enzyme it is. Hopefully you remember from cell and molecular or cell biology if you've already taken that with enzymes there's going to be an active site, a pocket where the substrate can fit in. And you notice up there, that little red and black is the substrate. That substrate has to have a specific shape, not only to fit in, but to interact with the amino acid R groups in the active site. So it fits in, it interacts with the R groups, and then causes that induced fit, the conformational change of the enzyme. Whatever fits into it only has a slightly correct shape. Well, it's not going to cause the enzyme to do anything because not all the correct R groups will be interacting. If not all the correct R groups are interacting, no induced fit, no conformational change, no cut, paste, twist, tweak, or whatever it was that enzyme was supposed to do. And this stereospecificity is highly specific. The enzymes, the receptors on our cells, in our tissues, can represent minute, very small differences. I mean, you look, was it L-aspartyl, L-phenylalanine methyl ester, better known as aspartame, okay? To the receptors on our tongue, that's sweet. Slight conformational change, you notice they're highlighted in red, L-aspartyl, D-phenylalanine methyl ester. One sweet, the other's bitter. You look on the bottom there. S-citalopram, R-citalopram. One works, the other doesn't. And you have to look really closely there. What's the difference? Well, solid wedge to the left there, the S. If you look at it, solid wedge. Where's the jagged wedge? They flip. One way versus the other. One way, that benzyl ring pointing back. The other way, it's pointing forward. And that carbon with the amino group at the end, that carbon chain with the amino group, is going the other way. Slight changes, big differences.